So today we've got two projects where we've had researchers from Kew and from Forest Research working really closely together, using skills from both organisations, and two quite different projects, but both looking at genetic and environmental effects and adaptation. And we've got a triple presentation to begin with, and then a double one afterwards with several of our researchers from each of the projects. For those of you who aren't familiar with the Centre for Forest Protection, it's a research collaboration which is funded by DEFRA. In phase one, we've been funded by DEFRA Plant Health, and it's led by Forest Research and Kew, but we work also with several other collaborators. And the focus is really on protecting the future of forests and using innovative science and interdisciplinary collaborative research. If you want to know more about the centre, you can have a look at our website where there's information on all of the projects, and also you can find out a bit about our internships. Last week we had all of this year's 6 interns presenting, so you will hopefully be able to see some blogs from them about this appearing in the next month or so. So I am going to hand over now to Domen, who is going to be starting the presentation on the project. How does British silver birch grow in different environments, looking at the genetic and environmental effects? So hand over to you.
Yeah, thank you. Good morning into the first webinar of this morning. We've decided within the project to split the talk in, let's say, 3 equal parts. I will cover some sort of overview of the project and focus on the phenotypic traits, and then Rômulo and Phoebe from Kew will be taking on with the genomic analysis. We've tried to assess British silver birch and how well it grows in different environments in Britain from the genetic and environmental perspective. We had several aims, but within this talk we will present 4 of them. We wanted to assess the performance of populations that were planted at 3 sites. We wanted to see and understand the genetic diversity of the silver birch, see whether phenotypic traits are actually heritable or can we predict them with genomic data, and finally identify genetic variants that are associated with environmental conditions. The map on the right shows the 29 provenances that were part of this study. They are represented by white dots, and you can see that they are quite well spread throughout the country. And then with the black dots, you can see the 3 trial sites. So there is one in Scotland near Loch Tay called the Drummond Hill. Then there is one in Norfolk near Thetford, and called Thetford, and one in south of Wales called Llandovery. They were planted in 2001, so these are adult mature stands. And we measured several characteristics, several phenotypic traits, some quite standard for forest research like DBH, and some quite novel like carbon discrimination. And we also collected samples for the DNA analysis. On top of that, we've collected samples from 157 plus trees of silver birch. So plus trees are trees that have some sort of favorable characteristics. They're usually quite straight. They're usually low in branching. They exhibit nice growth. And they are potential trees for any breeding program that we want to establish. So the meaning of this part of the project was to see whether we can find some sort of signal in the genome, whether these trees are actually being superior. So I think it's important to see the visual representation of the trial sites. you can see immediately that the sites differ from each other quite greatly. Especially Drummond Hill was on a wet and harsh site on a slight slope. So trees were basically throughout the years sliding down the hill and made some sort of banana shape growth, which was of course posed many challenges when we analyzed the data. Why do we want to work on provenance trials? Well, mainly because they're this amazing opportunity. They offer amazing opportunity to see the differences in different genetic material on site. So here you can definitely see the differences between the provenances that is planted on the left, which is quite thin, with the provenances planted on the right. So although they were planted on the same time and they have fairly similar growth conditions, they exhibit different growth patterns. The huge amount of time in the year 1 and part of the year 2 was spent on gathering all of the data, so on field work. Sometimes where we couldn't reach the fresh leaves for DNA extractions, we also used this quite finicky method to scrape the phyloderm below the bark, which is quite rich in good DNA. And then on the right, you can see how we measured resistance of the metal pin going throughout the trunk, which can be approximated then for wood density. And this method has been used for the first time in our department on such high number of trees. We've collected the following phenotypic traits. We've collected many more actually, but quite some of them, especially historical data, we were not able to pinpoint them to individual trees. So these are the traits that we focus our first paper on. I wanted to show this picture just to say that projects like CFP are not beneficial just for these direct scientific outputs, which are, of course, papers for us, but they also produce many, let's say, byproducts. In this case, we've developed our own software for analyzing resistance data from the microdrill, and it was actually developed by the CFP intern, so it's another benefit of having internship programs as well. What I wanted to show is that even though you collect some, let's say, data that is quite clear, we are not always comparing the same thing. So here we see on the left quite thick tree and on the right quite thin tree, and they are roughly the same dBH as the next two trees. But here you can see that one tree is growing almost horizontally in Drummond Hill, yet still being alive. And some trees die throughout the project, and these are all things that you need to take into the account. When we've done some sort of first exploratory analysis, we saw that if we order the provenances from from south on the left to the north by latitude, we've seen some sort of declining trend in the traits. This is for the dBH, but when we plotted also other traits that we looked at, we saw the same sort of pattern. So trees that were growing in the south on average had some sort of better performance in terms of the dBH, in terms of wood density, in terms of carbon discrimination, amount of carbon-13 isotope, which is some sort of proxy for water use efficiency. So they were more water use efficient. And finally, they were also taller. Not only that, there are several other aspects beyond the provenance that affect the growth. One of the very standard thing is the edge effect. So trees on the edge grow differently than trees within the stand. And using the models, you account for these differences, so your predictions are better. And finally, you want to look not only data from individual trees, but take into account some sort of broader population level. So just because trees are growing faster and seemingly better and they're healthier looking, it doesn't mean that they are more adapted, right? So here we see the mortality rates for different provenances growing in different seed zones. So seed zone 40, which had the highest mortality rate, is actually a seed zone that covers provenances from, let's say, South East England. And they had higher mortalities, especially when growing in the north. So even though they were looking healthier and they were taller, they also experienced higher mortality at the beginning of the experiment and throughout the experiment, actually. Once you take all of this into account, you can make multi-environmental models. And again, we've seen these clear differences, firstly, between the sites. So trees growing at different sites show different growth patterns. But also, again, this latitude effect of the provenances. So trees in the north, on average, had lower height, had lower DBH, but also were less water efficient, and they were having higher wood density. I will now ask the next speaker to take the control and continue the talk. Thank you.
Thank you, Domen. Yeah, my name is Rômulo. I work at the Royal Botanic Gardens with Richard Buggs and also Phoebe, who's going to be speaking after me. And we were looking more at the genetic side in collaboration with FR, who did what Domen has just described. So one of the things we want to evaluate was the genetic diversity of the birch populations we had genetic data for. And when we summarize the variation at the genetic level, we have this PCA here. If the populations are extremely different from each other, you would expect clusters of populations spreading in the axis of the PCE. But what we see is that most of the provenances, they fall within this big cluster here, suggesting that there's not a lot of genetic differentiation between the provenances, even though they're across the country. There are 4 provenances, 2 in the north and 2 more towards the center of the country that are actually a bit different. But when we investigated what was going on with those 4 provenances, we found out that actually it's not that they are genetically very dissimilar from the others, it's just that there are a lot of related individuals in these provenances, so sometimes even siblings. So we think the PCA is capturing some sort of family structure within these provenances. And we also identified the plus trees, which are in bright red, and it turns out that they fall within this big cluster, suggesting they have a similar genetic background from the other provenance. So the main conclusion here is that there doesn't seem to be a lot of genetic differentiation between the provenances in the country. So as I mentioned, when we did a investigation of relatedness, looking at pair-wire relatedness between individuals within provenances, we found many first and second degree relatives. So this kinship plot here is just individuals against individuals and how related they are, and the more yellow, the more related. So you can see that in kind of Chase 2, one of the provenances, you have a lot of clusters of yellow, which means that a lot of individuals are very related to each other. Whereas in Goldming, for example, you do not have that many. This seems to suggest that for some provenance, not all, the sample strategy was not as sufficient to get the whole of diversity of that population. So perhaps in the future, sampling more mother trees or mother trees that are more separated in space would be beneficial. One thing that we found was a bit, we were not expecting, is that some provenances had individuals that are related across provenances. So one individual be related to another individual in a different provenance, even though they were geographically quite far apart. So this could either mean that there's some, I mean, there were not many individuals, by the way. It was just, 5% of the trees had this kind of issue. And it was restricted to just a couple provenances. But that seems to suggest some sort of error during seed collection labeling. Or it could just simply mean that there was some planting and both provenances used a common source, for example, plus trees. So we cannot separate these two possibilities. But it was interesting from a logistic point of view. And also we found a few tetraploids, suggesting there was some downy birches as well in the data, probably because they look so similar. But again, these are a very small proportion of the data set. And then we moved on to looking at the genetic basis for the traits that Domen was discussing. So we were trying to find out whether there are genetic variants that are associated with the trait. So here, for example, we have carbon discrimination as an example. So this is what we call a Manhattan plot. Is just simply plotting the p-values of all those associations. We have millions of variants in the genome, so you're looking at each individual variable and whether it associates with the phenotype. So whether you have a copy or two copies of that variant, whether you have, let's say, a larger DBH or not. So each dot in this plot is a variant. This is a p-value on your y-axis. It's logged and negative, so the higher the number, the lower the p-value, the stronger the association. And here the chromosome, where that variant is located in the chromosome. I ran 3 models, and when you have a large peak, that seems to suggest there is a region in the genome that is strongly associated with that phenotype. And one of the models gave a lot of peaks, as you can see, which would be very promising, very exciting. But it turns out that this model doesn't take into account the structure in the data. And in this case, structure simply means this latitudinal effect that Domen showed, because Birch colonized the country from north to south. So there are a lot of variants that are present in the north that are not present in the south. So this ends up being picked up by the model. But when you control for that in the model, as covariates, for example, which are the other 2 models in the 2 different colors here, you can see that most of those peaks disappear. So it seems to suggest a lot of this variation that we're seeing here, they are just correlated with the phenotype, but they are not causally related. But still, when you control for that latitudinal effect, you still find 51 variants associated with some of the traits. This is not a lot. It seems to suggest that a lot of these traits are controlled by a lot of genes. They're polygenic, and the effects of each gene is very small, so it's not picked up as a significant effect in this GWAS. So why did I choose to show you a model that it's imperfect? Because this peak here, when we started looking into it, we realized it was a very thick peak, suggesting it was a very big region of the genome, around 400,000 base pairs, that was, somewhat associated with the phenotype, but it could be just the noise from the structure. And when we looked deep into it, we found out that it's most likely a genomic inversion. So inversion is simply a bit of the genome that gets swapped over. So it's in a different orientation from the other individuals. So if you plot the variability in that inversion location, you see 3 clear clusters, which we think are the haplotypes. Red would be the individuals that do not have the inversion. Birch is diploid, it has 2 copies of each chromosome. So green would be individuals that have 1 copy of the inversion, and blue individuals that have both copies. Even though the blue one here looks a very small cluster, we have actually around 800 individuals clustered here. So this also seems to suggest that this inversion is quite recent, because there hasn't been a lot of time for variation to occur in this inversion. Otherwise, you would see something like this, something more spread. And as you can see here, although it's a bit small, you can see that this inversion follows a latitudinal climb, and that's why it was being picked up by that GWAS. So the inversion is much more, the frequency of individuals with 2 copies of the inversion, it's higher in the north. So when we take we do some analysis taking to the account, trying to control for this latitudinal effect, because remember, individuals in the north are smaller, for example, on average. But when you compare individuals within the same provenance that have or do not have the inversion, we do find an effect in the phenotypes, particularly DBH, water use efficiency, and wood density. So it seems that even taking the latitude into account, individuals that have the inversion tend to have lower values for these phenotypes or for these traits. So it's something that's still tentative, but it's interesting in itself. We don't know exactly. We looked at the genes in this region. None of them is extremely clear. There are a lot of them involved in apoptosis, cell death, perhaps root morphogenesis. So maybe there is a link there, something we should pursue in the future. And next, we look at the heritability of those traits, just how much of the variation in the trait is actually driven by genetic variation. So the higher the value in this column here, it means that more variation in the trait is explained by the genetics. And as you can see, some of the traits have quite high heritability. So height, for example, has 0.62, which means roughly that 62% of the variation in height is due to genetics. Same with wood density, but others not so much. Carbon use is different at 0.3. Steam form has very little genetic basis, but that seems to correlate well with what we found in Domen analysis, that a lot of provenances which are genetically different were growing in different, achieving different heights no matter where they were planted. We also did a genomic prediction, so it's literally just a model, like you're attempting to learn the information from the genetics and trying to predict the phenotype of other individuals. So you try to see how the genetics relates to the phenotypes of a testing set, and then you select a different set training set, and then you use a test set to see whether the breeding value that you're estimating from the genetic data correlates well with the phenotypes of those individuals. And we can see that the accuracy of these predictions of the model are moderate. For height, for example, we have an accuracy of 0.58. And of course, when the heritability is low, the prediction is also very low. But these accuracies are, compared to the literature, are reasonably good. So that means that in the future, these models could be used in a breeding program for speeding the breeding cycle, because you don't have to wait for the trees to grow, as adults in order to predict how well they're going to perform. You can just look at their genetics from the model and see whether they have a high or low breeding value, right? And finally, we use those models that we trained with the provenance trees to estimate the breeding value of the plus trees. Because as Domen said, these trees were selected presumably because they have some merit with respect to their height or form. And what we find is that when we calculate the breeding value for those trees using the models, it turns out that for some traits, the breeding values for those trees are actually higher than for provenance trees are at similar latitudes. So if you look at this regression, you have latitude and the breeding values. The breeding values tend to decrease with latitude, which is, again, what Domen showed. Trees in the north tend to grow smaller, for example, no matter where they're planted. But if you look at, for example, at high latitudes here for height, the purple line is the plus trees. You can see that they have, on average, higher breeding values than trees from provenances at the same latitude, which seems to suggest that the selection for those plus trees was successful, that they are, if you were to plant these trees or offspring from these trees, they would tend to grow taller than picking individuals at random from similar populations. So now I pass the word to Phoebe.
Hi, I'm Phoebe. I'm just going to explain a little bit about the Genome Environment Association side of the project, which saw us associate the local climatic conditions of each of our silver birch provenances with their genomes to identify genetic variants that are exhibiting an association with ecological gradients. So we ran this analysis on 16 environmental variables, covering a range of classical temperature and precipitation climatologies, as well as wind and terrain. And through this analysis, we identified 218 genetic variants as associated with the environment. And as you can see from this bar chart, the majority of these were found linked to wind speed, but we also found variants associated with annual precipitation, water flow direction, mean temperature of the wettest and driest quarters, number of frost days, and TPI, which is topographic position index. And this actually covers quite a wide range of variables to which silver birch are having to respond to, highlighting these factors as key selection pressures requiring the greatest genetic adaptation. So as I mentioned previously, the highest number of associated variants were identified for wind speed, which in itself is quite interesting given that silver birch are a wind-pollinated species and their seeds are also dispersed by wind. Though of course wind can also be a stress for trees, it's responsible for uprooting and increasing rates of evapotranspiration. And upon closer inspection of the Manhattan plot for wind, which is very similar to the one that Rômulo was showing before, so each dot is a variant, the genome is along the x-axis, and then the higher up the variant on the y-axis, the closer its link with the environment. And we found that the variants were within these quite defined regions of the genome, and these are known as peaks. And these areas then show a very strong link with wind speed. And we can take the highest variant from each of these peaks and we can plot its frequency within each of our populations against the wind speed measured at the local conditions of each of those populations. And in some cases, we find the genetic variant to show a really nice trend where the frequency of the variant decreases as wind speed increases or vice versa. But in other cases, this pattern was a lot less prevalent, with the one outlying provenance with this much higher occurrence of this variant. And this leads to a much more spurious association. And this outlying provenance was actually seen across many variants and environmental conditions. So we think that perhaps something strange is going on with this population. So maybe it was planted rather than wild, which is leading to quite different frequencies than other even neighboring provenances. So one way that we can better characterize these associations is through annotating these variants and seeing which genes they are within or very close by. And by doing this, we identified genes such as endoglucanase and lacase, which are involved in cell wall strength and expansion, as well as phosphoglycerate mutase, previously shown to be involved in stomatal control, and aspartate aminotransferase, which is involved in energy production. So this just highlights these very specific genes as potentially involved in responding to wind, and therefore possible candidates for conferring greater environmental resilience. So in order to determine whether our silver birch provenances are locally adapted, we then looked at the suitability of each of our 29 provenances to the 3 trial sites that they were planted in. So we might hypothesize that provenances that are located closer to the trial sites in terms of geographical distance, but also ecologically, will show a greater suitability to that trial site if they are locally adapted. And these plots just show the differences in environmental conditions between each of our trial sites, which are shown by the triangles, and then each of our provenances shown by the purple dots. So these plots have just collapsed the variation across a whole range of environmental variables onto just 2 axes. And it becomes quite apparent that the conditions at Drummond differ quite considerably from each of our provenances. So it exists separate in the plotting space and it's outside of this dark grey envelope. And Drummond is actually noted as being this particularly harsh site with a steep slope and quite great exposure to wind and temperature. And so this might explain some of the differences that are shown here. On the other hand, Thetford and Llandovery have local environmental conditions that are much more similar to each of our provenances, and they are within this provenance climate envelope. So we might expect that genetic suitability of each of our provenances to Drummond is a lot lower than for Thetford and Llandovery because the provenances are locally adapted to a different set of ground conditions. So we then predicted the genetic suitability of our provenances to each of our trial sites. by using the 218 genetic variants that the GEA had found associated with the environment. And we predicted their frequency in each of the trial site conditions. So we can then compare the baseline frequency of each variant in the original provenance location to the predicted frequency in the trial site conditions. And the difference between these two values then represents the amount of genetic change needed for that provenance to be best suited or adapted to that trial site. And this is often called the genomic offset or the allele shift, but it just represents genetic suitability. And we performed this calculation 3 times for each of our trial sites. And we found that the amount of genetic change needed for our provenances to be adapted to Drummond was a lot higher than that predicted for our more southerly trial sites, to which the provenances showed very similar levels of genetic suitability. And as I said before, we think this may be because the local conditions at Drummond are outside of this provenance climate envelope. But we do see that this provenance here, Elgin, has a very low predicted genetic change required to be suited to the conditions at Drummond. And Elgin is actually located in Scotland, quite close to Drummond. And so this lower predicted genetic change and greater suitability may be because Elgin is more locally adapted to this more exposed and hostile climate. Another very obvious outlier is Prestine, with this very high predicted genetic change for Llandovery and Thetford. And this provenance is located within central Wales. It's fairly close to Llandovery, and itself, it's not an outlying provenance from a genetic point of view. So we're currently still trying to look into this to understand why this is occurring. So with the wealth of phenotypic data that was also measured over the course of the trials, we then compared each of our genetic suitability estimates to the mean performance of each provenance within each trial site. So we might hypothesize that a greater predicted genetic suitability would result in better tree performance. If an individual is locally adapted, it should know how to grow best. And this pattern was very marginally seen in Llandovery and Thetford, where there is a very slight negative trend with greater predicted genetic change seen at lower phenotypic trait values. So shorter, thinner trees with thin bark were seen when the genetic change required was largest. But this pattern was not significant. For Drummond, this pattern wasn't really seen, and DBH actually shows a positive pattern with greater genetic change seen for trees that are actually growing the widest. But as we've already established, Drummond is outside of this provenance climate envelope, so this might explain some of these more contrasting patterns given these greater ecological differences. But this has quite practical applications for the use of genomic forecasting techniques that are used to predict genetic suitability, because they don't seem to work as well when the climates being compared are considerably different. So here, although we're predicting that a larger genetic change is required for the trees to grow at Drummond, the trees planted there are surviving, and although shorter, actually have a larger DBH than trees in other trial sites. And whilst it could be hypothesized that this is due to different phenotypes providing a selective advantage in different environmental conditions, it's previously been suggested that environments that are outside of the species range, the genomic forecasting techniques are less applicable for the species that are outside of the range. So they need to be quite carefully considered when you're predicting suitability to current or future climates. So just to quickly wrap everything up, we found very little genetic variation between our silver birch populations, but the southern provenances were found to have higher performance across all 3 trial sites in terms of height, DBH, wood density, and crown density. Our GWAS analysis found that phenotypic traits were largely polygenic, with many variants contributing a small but cumulative effect to the overall phenotype. Our genomic prediction models were able to identify the preferential growth of the plus trees, validating this approach. We identified local adaptation to wind speed, precipitation, and frost, and genetic suitability to the northern trial site was predicted to be worse, but the trees are still surviving there and are still performing well. So finally, we would just like to thank everybody at Forest Research and Kew who has contributed towards this project. We'd like to thank the CFP for funding, and thank you for listening.
Thanks very much, Phoebe. So we have some questions. So if I can ask our three speakers to put their cameras on. So the first question is from Annika Perry, who said, really interesting presentations. For Domen, could the birch provenance that grew tallest but also had the highest mortality be demonstrating the risk of a grow tall fast strategy? Or do you think there's something else going on with these trees?
So, of course, we don't have like data to prove anything with this question, but I could speculate that this is what we had in mind. Yeah, so that's sort of provenances from the south basically continue growing, are just not used to the harsh conditions of the north. There is one very interesting aspect, though, where we found out that trees of the northern provenances tend to close their stomatas later, meaning that actually they have higher, let's say, growth period than the southern provenances, but the south provenances still grow faster. That's more of an interesting thing, but to answer your question with any sort of certainty, I sort of cannot.
Thank you. So the next question, this might be for you as well. So this is from Imam Syed. Does the material form the breeding population of the FTT qualified silver birch?
Yeah, so the plus trees that were analyzed are forming the FTT breeding population.
Okay, thank you. And do you know which populations are heavily presented in the orchard material, and are there any consequences?
Sorry, I do not. But if they are mostly from this FTT population, then we have nothing to worry about. They are either performing equally or slightly better.
Thanks. So the next one's probably for you, Phoebe, from Peter Jackson. What are the implications of the work on selecting the provenance of birch to cope with climate change?
I guess it depends on where you're going to plant them and what you're predicting your climate change to be, because there's lots of the like more practical, more like classical applications of the genomic offset work are to predict suitability to future climates. But I guess that because we're sort of ground truthing those sort of experiments where they're predicting this worse or best suitability to climates that are maybe not either going to happen or are too different, you might be over exaggerating to this like worse suitability, which isn't necessarily the case, as the trees in Drummond are still growing there and are growing, equally as well. And these phenotypes that we're seeing may be just adaptive to the harsher conditions at Drummond. So in terms of selecting provenances to cope with climate change, it depends where you're going to plant them and what you're predicting your climate change to be.
Yeah, so that would be perhaps some future analysis using future predicted climates, but using a similar methods to what you've done. Yeah.
I think ultimately, if you want to trust the genetic offset, then you just select those. You could run a model with just let's say the temperature variables and see which provenance would require a larger offset, and you would plant those that need a lower change. But I think our results show a more promising idea that, even the ones that require a big offset, they still can cope well with a harsh climate. So maybe you don't have to do anything.
Yeah. So there's actually lots of ability of those trees to adapt to the new climates anyway, with perhaps plastic responses. For you, Rômulo, from Rachel Riley, regarding the geographically separated birch relatives, so where you've got those related trees from different provenances which are interpreted as anomalies, is it possible that they're not planted but are relicts of natural populations?
Well, the thing is they're quite geographically apart. So how could siblings or half-sibs be present so far away from each other, or second-degree relations? So I'm from the top of my head, it doesn't sound like a natural thing. It could be simply that, yeah, they were just planted from a common source, or even easier, because this provenance trials are such a logistic nightmare. There's so many things going on. So it's normal that within any project, you commit a small mistake and label one bag with the incorrect provenance. But it could be just planting. And it's very difficult to find out. You'd have to trace back, try to locate whether something went wrong. It's very difficult to differentiate what's going on. But the important thing is that shows the importance of genomic work, because if you hadn't done this genomic analysis, you would never know that these things may have happened. So I think this seems to suggest that for provenance trials, it would always be good to have some sort of genetic component to the project, so you can tell these situations.
Yeah, thank you. I don't know if you can answer this one because I don't think you had it in your data set, but Jonathan Burton Rômulo is asking, did you find any influence of Betula, and I don't know if I'm going to pronounce this correctly, Celtiberica in the Welsh genomes?
For us to know where, let's say, we did find some triploids and tetraploids. They're not plotted in that PCA that I've shown. It happened before I took over, so I don't have that data. But for you to be able to tell whether other species are contributing to the genome of the ones in your data set, you'd have to have some representative of those species. So you could plot them in a PCA, and depending where the hybrids would end up in the PCA, you could infer where the differentiation is coming from, where the introgression is coming from. So I'm not familiar with that species, but if we had genomes from that species and we plotted them together in the PCA with the hybrids, maybe we could tell whether some of the individuals had introgression from that species. But the answer to your question is no, we don't know. We know probably, Betula, Downry Birch, probably.
Could you explain a little bit more, actually, for everyone who's not involved in genomics, when you're talking about the fact that you found tetraploids or other polyploids in the data set, what that means and how you know that that's a different species?
Well, it just means that some individuals have more copies of the chromosomes. In the case tetraploids, they have 4 copies of each chromosome, and we know Betula pendula only has 2 copies. You can run some analysis specifically. We're not going to get into this, but you can infer how many chromosomes they have. And we know that Downry Birch is a tetraploid, so it has 4 copies. So those tetraploids are probably Downry Birch that were misidentified. And some of them were triploids, so that probably means that silver birch crossed with Downry Birch and led to individuals with just 3 copies. So Downry Birch, dad offered 2 chromosomes, the mom, silver birch, gave 1 chromosome, and then they got mixed into it in 3. And it shows that even the triploids can survive, right? Because they were misidentified as silver birch and were planted and were growing fine. So yeah, it shows that hybridization, it's, if not adaptive, at least it's not damaging enough for the tree to survive.
Yeah, and once again, helpful to have the genomics work, because that's difficult to pick up in any other way.
Yes.
Finally, a last question from Tim Gordon Roberts. I'm not sure which of you wants to pick this up, but what are the implications for and we'll come on to this, Tim, in the next talk as well. What are the implications for policies that require planting local provenance?
Well, I think overall, it seems that the data is suggesting that it doesn't work for northern provinces, right? That you would, if you want, it depends what you want. If all you want is growth, if you're looking for timber, for example, all you want is a tree that grows very fast, then you wouldn't search for local provinces in the north. You would plant southern provinces, right? But then we would get into that discussion. Are the trees growing in the north smaller because that's an adaptive response for the harsh conditions, and that's just plasticity? Then in which case you could still plant local if all you want is just preserving the diversity of that region. But if it's for timber, then no, you would most likely go for southern provinces. Domen, do
you have anything to add to that, Domen?
Yes, I would agree that you can conservatively use some sort of rule of few, like 3 degrees from the south and plant it 3 degrees north. But when you have like a known harsh environment, I wouldn't gamble with survival and would plant something more local. So when you have like exposed areas, areas with late frost and stuff like this, I would definitely plant more adapted local material.
Right. We're going to move on now to our next project, and we've got Guillermo from Kew and Nikki from Forest Research, who are going to be talking about their joint project evaluating genetic diversity in planted and naturally colonised young woodlands. So I'm going to hand over to them.
Thank you all for taking the time to be with us today and taking an interest in so many of the projects that are going on. Some very interesting work. As Louise mentioned, my name is Nikki. For those of you who don't know me, I'm based at Forest Research, and I'll be presenting alongside my colleague Guillermo from Kew Gardens. And as with the previous presentation, if anything is unclear and requires more explanation, can you just pop a message in the chat so that we can address it? So we'll get started. As we are all aware, there is a global initiative established to expand forest cover as a means of safeguarding biodiversity and mitigating climate change. Tree planting has become a widely adopted strategy to achieve this goal, supporting the natural processes of forest regeneration. We were interested to see if tree populations of English oak and silver birch were being represented by a diverse range of seed sources, as we believe that that will improve the genetic diversity within populations, in turn perhaps improving tree susceptibility to pests, disease and climate change. If there is a decline in genetic diversity and inbreeding occurs, this could lead to potentially unsuitable tree species to help with the threats they are likely to be facing in the future. So the aim was to compare the levels of genetic diversity in planted versus naturally colonised forests using a lot of genetic data to try and identify potential population bottlenecks by loss of genetic diversity and or selection across age cohorts, whilst also considering differences in health indicators using statistic modelling. So I'll spend a few minutes just going over the project and methods, etc., but I wanted to outline some of the summary findings. So we will revert back to these findings at the end of our presentation, but I wanted to go over what we'd summarised. So we initially found, based on information obtained from forest reproductive materials, it seems that for more than half of the 39 UK tree species analysed, seed collection appears to significantly underrepresent the range of climates in which those trees naturally occur. This probably needs to be taken with a pinch of salt, as it might not include all the stands and might have correct geographic locations, but it's what we found from our initial investigation. We also found that overall genetic diversity metrics for planted and naturally colonised are quite similar, which is good news. It means that the planted populations can emulate the genetic diversity of wild populations. However, on the other side of this, the planted populations were more similar to one another than, say, a naturally colonised set, particularly for oak. We also find weak but significant evidence that there is selection in nursery pipelines, so we may or may not be introducing biases to the cultivated material. So what we're looking to compare. So current afforestation strategies in the UK to meet conservation goals can be broadly classified into two main types. So afforestation pipelines based on natural colonisation or regeneration, where tree communities establish in, say, former arable or farming lands by natural dispersal. And then we have the afforestation pipelines based on active planting using commercially produced trees. So each strategy involves different processes that may affect the levels of genetic diversity in resulting woodlands. So parental trees in wild populations, for example, may arguably represent a wider genetic pool and be better adapted to the target site than those in geographically distant seed sources or orchards. But in turn, selective pressures across age cohorts, such as climate change, predation, pests, diseases, may reduce genetic diversity through differential mortality in natural populations. Conversely, planting protocols that integrate multiple seed provenances and aim to maximise germination and survival through highly favourable growing conditions can result in more diverse populations than natural ones, in theory. It is possible that these populations, through natural selection, are better to keep pace with emerging stressors. However, commercial seed collection, germination, nursery cultivation, and/or planting protocols can also potentially inadvertently introduce genetic bottlenecks or selection at one or more of the life stages. So in this study, we focus on 3 different age cohorts across the natural colonisation and planting pipelines to test these competing hypotheses. So sites were selected based on their planting and colonisation of the target species. So we were interested in oak and birch, and we did this on former arable land, with existing woodland located nearby with mature trees that are seeding and therefore the potential trees for the colonisation. It did prove to be somewhat difficult, as we know birch seeds from such a young age. We obviously didn't want the natural colonisation to be seeded from the planted populations. So without going into too much detail, I'll hand over to Guillermo now, who will give an overview of the results from the genetic side.
Thank you, Nikki. I hope that you can all hear me well. So yeah, as Nikki already mentioned, and before analyzing genetic differences between oak and birch afforestation strategies, our first aim was to test if the variability in environmental conditions of the tree populations in the wild were sufficiently represented by seed sources. So our aim here, the idea behind this test is that if different wild populations are adapted to different conditions, but we rely on only a few of them as seed sources, we can lose some of this adaptive variability in planted populations. To understand these potential environmental differences between wild populations and seed sources, we first mapped registered seed sources stands obtained from the UK Register of Basic Materials, as well as from master certificates when they were available, for a set of 39 UK native species. You can see some of them as an example on the left panel of the slide. The second thing that we did was to extract climatic values for all the variables listed on the right at the seed sources for each one of the species, as well as across their wild distribution. And then we tested for significant differences between these 2 sets of data. So what we have here on the left, what you can see here is a spatial representation of the climatic condition of seed sources in green, and of the climatic range of wild populations in orange, based on a principal components analysis. A principal components analysis is basically an approach to summarize variability in a large data set with many variables to be plotted into a bidimensional plot like you have here in the slide. So this is not a geographical map. Each dot represents an individual or a data point and occupies a location in the plot depending on environmental scores measured where they occur, yes? So the important thing here is to notice how the wild populations consistently cover a broader range of conditions than the seed sources. And this was the case for 23 out of the 39 tested species. These differences were significant in 23 of all the species tested, including the 2 species of interest in this study, Betula pendula and Quercus robur. So this suggests that seed collection often overlooks the full environmental variability found in the wild, which may reduce the adaptive potential and resilience of planted populations to current climatic conditions, and also those changing in the future. It is important to note, though, that this analysis is based on the UK Register of Basic Materials, which likely doesn't include all the stands used for seed collection. With more complete records, we should be able to test this pattern in greater detail and with higher accuracy. Okay, so moving on to the genetic analysis on oak and silver birch, on pedunculate oak and silver birch. Let me tell you a little bit about our sampling design. We sampled around 25 individuals per cohort. The cohorts that Nicky already mentioned, parental trees, seedlings, and saplings, from 4 wild populations where natural colonization was ongoing. For each one of the species, you can see the locations in the map in the slide. For planting pipelines, we sampled trees from 3 planted sites, also shown in this map. We sampled also seedlings from different origins, from 2 nurseries, Mylord Forest and Cheviot trees. And in the case of birch, we could also sample a seed orchard. To complete the data set, we also included samples from natural stands representing seed sources. And in total, we obtained genetic data for 459 oaks and 625 birch trees. So with this data, the main questions that we wanted to answer here were, first of all, is genetic diversity in planted pipelines higher or lower than naturally colonized populations? So we have 2 competing hypotheses here that need to be tested. In the first scenario, seeds sourced from a limited number of parent trees can result in populations, planted populations that have lower diversity compared to wild ones. But also, on the contrary, seeds sourced from multiple provenances and mixed may actually result in populations that have higher diversity than reference wild populations because of this multiplicity of origins. In second place, we wanted to test if planting pipelines can relieve inbreeding compared to naturally colonized populations. Small and fragmented natural populations may be at risk of inbreeding and genetic drift, and planting pipelines done in a particular way may actually help to reduce this inbreeding in forested populations in theory. Third, we wanted to test if planting pipelines generate sites with more similar genetic backgrounds than natural ones, or in other words, we wanted to test if planted populations were more similar, genetically speaking, to each other than natural ones. And this could happen through different processes. On the first scenario, using the same seed sources and multiple sites can obviously generate homogeneous planted populations, right? But using seed mixes, using the same seed mix in multiple sites can also generate populations that are more similar to each other, even if they are more diverse, as shown in the figure. The opposite scenario will be using site-appropriate seed sources, which can help to maintain population differences. And finally, we wanted to test if planted pipelines were introducing biases, were introducing selective pressures through the process. Seed source selection, nursery cultivation, and planting can actually introduce these biases, this selection, which is something that we can also test with genetic data. We started by testing for differences in genetic diversity using several indices. So each one of these symbols here in the y-axis are basically different ways of measuring genetic diversity. Each bar represents a population or group within each cohort or life stage shown above the bars, and for each one of the afforestation pipelines. So as you can see here, we found very minor differences when comparing groups corresponding to natural colonization and planting afforestation pipelines. The differences were non-significant for almost all of these comparisons. So what we can infer from this in regard of our first question of interest is that first, we don't have lower diversity due to limited seed sources compared to wild population. And second, we don't observe either higher levels of genetic diversity in planted populations as a result of mixing seed origins or because of variable conditions in the first stages of their life. Next, we tested for inbreeding across cohorts in both wild and commercially produced trees. Inbreeding measures the potential genetic loss, of genetic diversity as a result of mating between related individuals. Using a statistic called FRO, we found that most wild and planted populations had similar levels of inbreeding, except for natural colonizers. This is the seedlings and saplings that we sampled in recently colonized former arable lands. So on the plot, what you can see here is that the populations corresponding to the planting pipelines had low levels of inbreeding, as well as adult trees in the natural colonization sites. On the contrary, we had the colonizers of these arable lands, former arable lands, showing higher levels of inbreeding, and this pattern was repeated for both species. The truth is that during fieldwork, Nikki and I often observed that adult parental trees in natural colonization sites occur in small isolated patches. In some locations, I was even able to sample every neighboring adult tree because they were so few. This is so unusual that I was able to sample every individual in the populations. So this reflects how high is the risk of inbreeding in these populations. These results suggest that the planting pipelines can indeed effectively prevent inbreeding in afforestation programs, especially when seeds are sourced from orchard or from large mature forest populations rather than these small fragmented stands that we saw in natural colonization sites. Okay, next, we tested whether planted populations were genetically more similar to each other than natural populations, and we did so by calculating co-ancestry scores, which measure how closely related individual trees are. High co-ancestry is not the same thing as inbreeding that we explored in the previous slide. Inbreeding essentially estimates how closely related an individual's parents are, while co-ancestry basically measures how closely related I am, for example, to somebody in my populations of individuals. So the key connection between these two concepts is that a population with high co-ancestry is always at risk of producing inbred offspring in the subsequent generations. And that's the risk that we are measuring here. Co-ancestry scores can be calculated within a single population or between different populations. The results for this analysis are shown in these two heat maps for oak and birch. So let me explain a little bit about how these plots work. What we have here, so it's basically the same populations or group in rows and columns. So each cell represents a comparison in between two of them. Along the diagonal, we have a comparison of each group with itself. So the diagonal is a within group co-ancestry score, and the remaining cells are the pairwise comparisons. Let me remind you a little bit of the structure of our dataset here. We have 4 natural colonization sites for each one of the species with 3 different cohorts: adults, seedlings, and saplings, colonizers. And then we have the populations or groups corresponding to the planting pipeline. We have seed sources, an orchard in the case of birch, nurseries, and outplanted populations. There is a lot of information in this plot, so let me guide you through the key findings here. The first pattern that we observe is that the highest levels of within group co-ancestry occur in the populations or group corresponding to the planting pipelines. So we have these cases here of nurseries and outplanted populations in oak, and basically almost every group in commercially produced trees in the case of birch. And this is problematic, right? Because as I already mentioned, high co-ancestry can result in inbreeding in subsequent generation, and that can also result in a loss of genetic diversity. There was an exception to this pattern of heightened co-ancestry in commercially produced trees, which was the only orchard that we had in our data set. And this is encouraging because this suggests that orchards can be a good strategy to avoid this problem, this risk of high co-ancestry and subsequent inbreeding. The second pattern that we observed is that co-ancestry scores among populations in wild populations in natural colonization sites, basically those comparisons within that triangle, were lower in natural populations compared to comparisons between or among nurseries and oak-planted trees, which are represented within the lower triangle. So this indicates that there is, to some extent, to some degree, some genetic homogenization going on in the case of oak. And this is probably due to the fact that the vast majority of seeds used in the afforestation of oak in our data set came from a single seed zone, the zone 405. We didn't find this pattern, or at least not so pronounced, in the case of birch when we compared the two groups. And this is most likely because in the case of birch, there was way many more origins of seeds used for planting, which helped, it looks like, to maintain genetic differences among populations. So in summary, planted pylons can indeed lead to more similar genetic backgrounds as we see in oak. However, using diverse seed sources, as in birch, can help preserve genetic differences between populations. Such is the goal, for example, of favoring local provenancing. We could not test if seed mixing can also result in more similar populations because we only had one such population deriving from the birch orchard, so comparisons couldn't be made. But at least we found this pattern, this result of absence of heightened co-ancestry scores with respect to wild populations. So the last question using genetic data that we wanted to answer here was whether selective processes are occurring in planting pipelines. For this, we focused only on colonizers and unplanted trees. We excluded adult trees and those from nurseries because we wanted to see if being planted or naturally colonized explain to some extent genetic differences that could occur between these two groups using a statistical method that is known as redundancy analysis. So to illustrate this analysis, think of a group of people from which we collected height measurements. If we group them by nationality, let's say these people are just Dutch and Spanish, we will most likely see a significant average difference as the Dutch are the tallest people in the world on average. But if we classify them based on a less relevant variable, like a preference for the beach or the mountains, we will probably not see a meaningful difference between the two groups because this variable is most likely not informative of height, okay? So here, our explanatory variable is planted or not, and instead of heights, we have genetic profiles, we have genotypes. As these plot results show for oak and birch, colonizers in orange and planted trees in blue do not overlap. They do show differences. And this clear separation between groups indicates that the variable planted or not is indeed relevant. Or in other words, this indicates that being a colonizer or a planted tree is associated with genetic differences, which in our analysis were limited but significant. This analysis also allows us to identify which specific genes along the genome show greater differentiation. In our test, some of these genes had functions particularly relevant, such as genes influencing flowering time or for how long seeds stay attached to the tree. These are traits that could be unintentionally selected during seed collection, for example. So, do planting pipelines show evidence of selection? Yes, we found a clear signal of selection. However, further research is needed to understand the strength of these signals and their effects on the resilience of planted forests, because as it is at the moment, we ignore if and to what extent these potential biases in planting pipelines, in planting protocols, can impact the sustainability of resulting populations. Nikki will now present data on health indicators, on pest incidents, which also suggests that planted trees and colonizers may be facing different selective pressures.
So yeah, so not only did we look at the genetics, but we also considered the phenology of the naturally colonized and planted trees. We were interested in assessing the main pests and diseases associated with birch and oak, such as oak powdery mildew, leaf spot on birch, galls, leaf rollers, crown health or crown dieback. We also visited the trees in winter to assess their form, as indicated in the diagram on the right-hand side. All of these variables that we assessed are associated with causing the tree some form of stress by either affecting its photosynthetic capabilities or causing stunted growth, crown dieback and such. So what did we find? Overall, the trees are actually quite healthy, so that's great news. There were few differences observed between planted and naturally colonised. Now, we can surmise that as most of the trees at the sites where comparisons were made, the youngest were already 10 years old, +2 for the planted ones that were grown in the nursery initially. As we're comparing trees of the same age for health assessment, they are probably past an age where they are considered to be vulnerable. But obviously, this was just the sites that we could find and the age of trees that were there. Most observations were visually recorded, so we also performed an assessment via chlorophyll fluorescence, and this also supported the visual protocols that the trees were found to be in good health. Statistically, we did find that there is a lower likelihood of mildew in planted oak, and there is a higher likelihood of leaf spots in planted birch associated with the fungus Myricinina betulae. Obviously, I went over the summary results at the start, so we're going to go back to those now. So as Guillermo went through, for more than half of the 39 UK tree species analysed, commercial seed collection significantly underrepresents the range of climates in which those trees naturally occur. So, for instance, we're collecting seeds from seed zone in Scotland and then planting in the south. We don't fully understand the impacts of doing this. The trees all appear to be healthy, but should we focus more on where the seeds are being sourced? We record the seed zone so that we can look at the provenance, but what about specifically planted trees from, say, seed zone 405, which is where the majority of our oak was from, on the same plane as the target planting site? So there are lots of questions that we still want to answer and wanted to discuss. Could we increase the number of parent trees? Seed zones are quite big if you look at the FRM map. Can we get this more knuckled down to what 3 words or GPS coordinates perhaps? If we only use registered stands, are we then limited? Do we need to get more stands and increase the genetic variability? Obviously, we don't want to make the processes more complicated. Nurseries find it as complicated as it is. So how can we do all of this to make it easier for seed suppliers and nurseries? As Guillermo said, the overall genetic diversity metrics for planted and naturally colonised are quite similar, which is amazing news. But there was evidence to suggest inbreeding in naturally colonised seedlings and saplings, so potentially due to a limited number of adult seeding trees, which highlights the importance of planting. But what about the naturally colonised trees? If we aren't using suitable planted trees, then what could happen in the future with the trees that have naturally colonised with limited genetic diversity, perhaps? Again, just something to think about. By contrast, co-ancestry showed that planted sites were more similar to each other than a similar set of naturally colonised sites. This was in particular for oak. Well, what does this mean? So basically our planted sites in the east are therefore genetically fairly similar to those in the south. So the same trees are being used for seed sources, which makes sense when you think about it. Nurseries outsource a lot of seed collection, unless they have their own orchards. And if you were collecting seed and you knew where the best seeding trees were and when, you would go back to them. You wouldn't waste time looking for new trees. But this means genetically our planted trees across the UK could be quite similar, and we don't know what impact this will have in the future. It might be fine, we don't know. This is obviously a sensitive subject as well. It's difficult to approach because obviously we don't want to say that one person's doing it right or wrong. with a fairly limited number of seed suppliers in the UK, how could this be improved moving forwards? We find significant but weak selection in the nursery pipelines. Again, this is understandable, but if we are selecting saplings for planting that are, say, for example, all the same height or of a certain root collar diameter, this could lead to a selection of functional traits associated with flowering time and acorn detachment, for example, which might not therefore be adapted to where they're being planted. Obviously, thinking of a worst case scenario. There might be a loss of genetic variability or functional traits that could be useful in the future. Again, with birch, we found the number of planted trees to have significantly higher infection rate with the leaf spot, although this could be due to a result of being planted next to each other and the stand density is much, when they're much closer. So everybody that's been involved, there's been so many amazing people involved in this project, and this isn't even half of them. We've really appreciated everybody's help. I know some of you are here today, got some private sites as well that, you know, we're very open to letting us go on and do lots of sampling. We did do the phenology over 2 years as well. So we went and visited them 2 summers in a row and 2 winters. And yeah, everybody that's been involved has just been amazing. So a huge thank you to everyone and whoever's here today. And I can see there's lots of questions, which Louise, do you want to mediate?
Yeah, yeah. Thank you so much for that, both of you. That's fantastic. Yeah, we have got quite a few questions. So, very first one is from Alex Pritchard, who said, in your investigation, is there an increased trend in hybridization of Quercus petraea and Quercus robur? This can be seen in a number of oaks close to Rydal in the Lake District, and could this be provenance related?
Actually, maybe Louise, you are better equipped to answer this, right? Based on your investigation. The thing is that because it's so easy for Petraea and robur to hybridize, we used in our analysis a reference genome that is a hybrid. So even when we try to target a more consistently Quercus robur, there may be some hybrids or even Petraea in our data set. There may be the least. But we don't look into patterns related with hybridization. So I cannot really comment a lot on that. So maybe Louise, you can actually.
Yeah, so I'll add to that, Alex, and do feel free to contact me if you want to, because I did part of my PhD on hybridization, and where you are in the Lake District is an area where natural hybridization is very common. And so it's more likely to be natural than provenance related, I would think, but I'm happy to talk to you about that offline at some point. A question from Joe Clark. So Guillermo, I think you mentioned that there was a sort of limited number of parent trees, and she's just saying how many would you consider to be a limited number?
Well, the thing is that we say that there may be a limited number of parental trees based on our data, based on our data set, which obviously it's very difficult to capture all the ongoing variation going on in all the plantings, but based on what we saw. We say that there is a limited number of parental trees compared to wild populations. That's our reference. So if I had to go for a number, I couldn't say a number of trees particularly, but something that actually reflects or emulates what we see in natural populations. We infer that there is a reduced number potentially in the case of oak of parental stands, basically because the co-ancestry among planted population is higher when we compare planted populations with each other than when we compare natural populations. So there is, I mean, based even in our results, it's difficult to make a specific recommendation in that sense, because if you favor, for example, local provenance, and if we limit our seed collections for planting to a seed zone, obviously you have more chances that you're going to have higher co-ancestry in that group, right? I guess it can even be, there's room for improvement possibly even within a single local provenance. In the case of UK, and this may be a bit well far-fetched, but the thing is that we've seen in many different species in the UK of trees that the population structure is relatively low or low. So maybe in the case of the island of Great Britain, the local provenance, favoring local provenance can be not that favoring, not that positive, and including seeds from further regions, if that results in lower levels of co-ancestry, that could be actually a right way to move ahead. But even within a single seed provenance, for example, the seedable co-ancestry patterns could be at least those that are similar to what we find in the wild.
Thanks, Guillermo. And another a question from Annika Perry. What age was a sapling?
So the thing is that, first design, when we were being very, very optimistic, we wanted to separate the seedlings and saplings based on basically rules of thumbs to classify seedlings and saplings, which I don't quite remember now, but it has to be with a particular DBH and a particular height, right? But we realized that it was impossible to actually make to actually build similar groups in terms of numbers to be compared. So in each population, basically, we divided each set of colonizers in two based on DBH and height, but they're basically split in two based on those two metrics. So planting, well, the year in which these former arable lands were left to regenerate ranged from 2011 to 2019, if I recall well, Nikki. Maybe you know about this better than me. But yeah, we have different ranges of age, different ranges of classification into seedlings and saplings.
Yeah, thanks. So I think the reason for that was Annika said it would be interesting to also test trees which are reaching reproductive maturity, as you might expect the inbreeding coefficient to reduce as the trees age due to fitness costs for inbred individuals.
Yeah, at some point it will be awesome to be. There was we had some problem. So our idea here was to be able to compare stages along planting pipelines against what we see in the wild, right? So we had in planting pipelines, we have seed sources, we had nursery trees, and we had outplanted trees in their early years. We could have gone further than that, but we selected those three categories or age classes so we could compare the two afforestation pipelines, basically. But yeah, looking forward into adult planted trees or adult regenerated trees, that would be nice to do.
Yes. Yeah, so the nature of the sites meant that wasn't feasible. One, Nikki, you might be able to answer. The FRM requires an Ordnance Survey grid reference for every collection, and could you use this to refine seed source information?
Yes, but obviously we were limited into what we could access, and we're also relying upon that being correct. I mean, dependent on the seed collectors, they're just collecting from a certain region and just giving a coordinate, but it could be from like 20 miles away. It's not as accurate as we hoped it would be. So, but how we're supposed to moderate this, how anybody is supposed to find that exact information out is one for the future, I think. But
thanks,
yeah.
Nikki. Domen says, this is probably for you, Guillermo. Wouldn't you expect that levels of inbreeding in naturally colonised areas, especially fragmented ones, are to be expected and therefore not considered problematic?
The truth is that we didn't expect it. This was a result that we just found, and we thought it was so interesting. Yes, I guess if I had thought about it before actually running the test for this particular result, I could have guessed, but we were possibly more inclined to think that maybe bottlenecks in planting pipelines could result in inbreeding. The truth is that this statistic, and actually every statistic that measures for inbreeding, can also be a proxy for genetic drift, which is not exactly the same process. Genetic drift affects small populations in which variability is lost due to random processes that don't really occur or not so seriously when the populations are big, because the alleles are more easily maintained in big populations. So I guess if I had, yeah, we could have a hypothesized from the very beginning that these small patches surrounded by former arable lands or still agricultural lands could result in this pattern. That said, I mean, that doesn't make it less problematic in the sense of if we're going to relate in natural colonization for seeding, that could be positive if seeding populations, the populations of adult trees in this natural colonization site were large mature trees, but that's not the reality of the landscape, right? Not in countries like the UK or any country, for example, with this level of development across the landscape. So I think it can be actually problematic in the future if we only relied on that, and also like targeted planting, assisted gene flow can help to prevent this.
Thank you. So a follow-on question from me on that, and I can see Jeanette Hall has also put a comment on this. But so bearing in mind that both the species you're looking at are sort of quite widely distributed and they're wind pollinated, and we know from other studies that there isn't a lot of population structure in them. Do you think there's something really specific about these agricultural sites that might make the results different from a more continuous distribution of woodland if your samples were from elsewhere?
You mean in terms of the trees that are established in those areas, or?
Yeah, in terms of the inbreeding scores, you know, because these sites are perhaps more fragmented than other sites where these trees might be found.
Yeah, no, obviously, I mean, this is, again, I wouldn't say speculating because the results are kind of clear, but this is our hypothesis about it is that inbreeding is due to the small size of these populations that are sitting these areas, and if regeneration or natural colonization happens in other areas that are more afforested, where seeding forests are larger and more mature and not subject to drift, maybe we wouldn't see this pattern. But the truth is that this fragmented setting of seeding trees was predominant in all the populations that we visited, and we visited a lot of them. Not only the ones that we are showing here, I think we visited between Nikki and me nearly 50-55 populations. Some of them were discarded because of a number of reasons that didn't fit our requirements. But in terms of what kind of land use from before, we suspected that there can be an influence, and that's why, to control for those potential differences, we focus only on one type of land. That doesn't mean that there are no differences. We found difference in terms of a slope, of drainage, but at least we wanted to control for that. So potentially yes, but we don't know really if that implies a difference in the trees that get established, also in terms of inbreeding.
Thanks, Guillermo. Right, next question. Fraser Bradbury says, it's interesting to see the limited seed sources for oak in the UK. Do you know how the introduction of European source trees affected the genetic diversity, as we've been using European seed sources for decades, if not hundreds of years? And yeah, the seed sources from anywhere it can be collected if there are poor masting years.
Well, we actively avoided populations that were planted with seeds that were not from the UK, because that's what we wanted to test. And that was also very limiting, because in many cases it was very, very difficult to track down the origin of the seeds. That was Nikki's work, really. She did an amazing job on that. So no, we don't have data on how the use of seeds from outside the UK, from mainland Europe, may affect the diversity in plant populations. But it's true. I mean, the landscape, the forests are so transformed. There's been planting for many years, centuries even. So there is most likely a signal of that, even in our data set, but we didn't test for that particularly.
Okay, thank you. Jonathan Norris asks, as there's no major difference in genetic diversity between planting and natural regeneration, does this bode well for adaptation to climate change? I.e., whether we want to plant from 3 degrees south or locally adapted natural regeneration, they both stand an equal chance at successfully adapting.
Well, that's something that we may be able to answer better in the next phase of our project. We just got an extension to test, similar to what Phoebe was showing before, these levels of maladaptation, potential maladaptation, or genomic offset, when basically you compare the correlation between genotypes and environmental variables. From what we see, and also this is something that came out in our previous meetings with the stakeholders that we had in the previous few weeks, there is a sense of that there is not so much phenotypic variation latitudinally, although Rômulo and Domen just show the opposite. In terms of how diverse planted populations are, I would say that I can only speculate about how beneficial may be to actually loosen or relax those restrictions, and maybe allow for more seed mixing, for planting seeds from other seed zones. The seed zonation system itself, it's maybe not super informative in terms of environmental variability. So maybe there are better ways, and maybe this 3 degrees latitude rule is better than, for example, seed zonation for seed sourcing.
Yeah, so that probably relates to this next question from Jenny Wong, who says, how do your results relate to the identification of GCUs for these species?
Again, so this is something that probably we be able to answer better in the next stage of the project. We are not yet there. We found some genes that could be under selective pressure in forestation protocols, and some of them can be relevant for even ecological adaptation or local adaptation, but we don't know for sure yet.
Brilliant, thank you. There'd also been some questions about Centre for Forest Protection papers. So if you keep an eye on our website and sign up to our newsletter, you'll get notifications of anything that comes out like that and any summaries. Jeanette, yes, the pest and pathogen webinar last week was recorded, so get in touch with me. So I want to say a really huge thank you to all of our speakers today. It was really fascinating talks, and also to our audience for attending, and also for asking so many interesting questions. It's really brilliant to hear from so many of you. Thank you, everybody.